Hello. Good morning, everyone. Welcome to this Sunday virtual Chai and Wine on behalf of TIFR. This time it will be hosted in St. Xavier's College, Mumbai. So we are very happy to host Chai and Wine this time on 30th November 2025. So today's speaker will be Ms. Divya Gohil. She's from TIFR. She's from Dr. Geeta Vaidya's lab. She's a DST Inspired Fellow and working in Dr. Vaidya's lab. Our topic for today will be why your immune system doesn't attack you. I think this is a very pertinent question, which the first immunology question that in the immunology class, the question will be why your immune system doesn't attack you. So she's going to throw some insights on this topic today, which is the Nobel-winning discovery of regulatory T cells. So we welcome all the participants, we welcome the speaker, and we welcome the PFR representative, Ms. Ulhas. And over to you, Ms. Divya Goyal, for the talk on why your immune system doesn't attack you. Thank you.
Thank you so much, ma'am. First of all, I'd like to thank all of you to take a time out on a Sunday morning, and it's a fairly good amount of people here. So thank you so much for being here. And I'll be specifically talking about the Nobel Prize discovery this year. And before I go there, the presentation is going to be like a little bit of understanding how our immune system works. And then we'll see how the scientists came to make this discovery. Ulas, can we have the presentation? Perfect. Okay, let me do like quick checks. Okay, so before I begin, can anyone tell me what you all understand when I say autoimmune diseases? Anyone, like what comes to your mind when I say that someone has an autoimmune disease? Absolutely no idea, or you all have some idea? Two? Sort of there, but autoimmune, guys? Harsh? Perfect. That's precisely what an autoimmune disease is. So, autoimmune diseases occur when your immune system makes a tragic mistake of identifying your own cells as if they were some foreign invaders in your body. Okay? And one second. Let me go back. Yeah. So when this misidentification happens, you develop a lot of autoimmune diseases like type 1 diabetes, where your immune cells start attacking your pancreatic cells, or you can have a neuro complication like multiple sclerosis, which attacks your insulation around your neurons, or you can have something like rheumatoid arthritis where your joints get targeted. The Nobel Prize discovery of 2025 in physiology and medicine honored these three scientists, Mary E. Brunka, Frederick Ramsdell, and Shimon Sakaguchi, for their discovery of special types of T cells or immune cells called regulatory T cells. These cells precisely prevent these autoimmune attacks in your body, and this is what we'll be talking about. Now that we've introduced ourselves to this topic, now let's just step back and look at how our immune system works. Okay? So our immune system isn't just one organ. It is a complex network of organs and cells, right? It involves organs like your bone marrow, your thymus, spleen, countless number of lymph nodes, right? And you have a whole army of circulating immune cells that work to keep your body healthy and fight against infection. So, but today, the focus of our talk is going to be about T cells. Now, the journey begins in the bone marrow, where all your immune cells are formed. Also, your T cells are formed again in the bone marrow, but they don't stay in the bone marrow. They travel to an organ which is a very small butterfly shaped organ above your heart called the thymus. In the thymus, these T cells get trained how to recognize threats and eliminate them. It's like a training school for T cells. So, what does our immune system actually do? We all know it protects us from a lot of dangers, be it virus, bacteria, pathogens, parasites. And our immune system can recognize these foreign particles, and this function of the immune system which helps it separate from the foreign to self recognition is what makes it powerful, which avoids itself from acting on our own body, but also eliminates these threats that we face on daily basis. But our immune system doesn't just stop there. It also identifies some sick and dying cells in our body, and it will clear these dying or abnormal cells like your cancer cells which are abnormal. The immune system also looks out for these kind of cells and clears it from our body. This is a small video where you will see, just can we click on that? Yeah. So in this video, you're going to look at a dying cell, okay? And then you will see these two immune cells coming and eating it up, right? And they're clearing this debris from your body. So that's what your immune system does. But the question is, how are these T cells recognizing what is foreign and what is threat? And this they do with simple signals called these abnormal molecules that are there, that are presented by either the virus or the bacteria or your abnormal cells. Now, these scientists call these antigens, okay? So your antigens act like signals in your body, so when your T cell is circulating in the body, it will keep scanning for any such foreign antigens in your body. And once it detects a threat, it will alarm the other immune cells in your body to start reacting towards this threat. Okay? So, okay, fine. Your antigens act as signals, but you need some detector, right, to detect those signals. So what are that detectors on your T cells that, can identify these antigens? So these are special proteins that are present on T cells called T cell receptors. And imagine they are like your lock-in key, right? It's like a very unique key. And when this key fits like a particular lock, the T cell gets the idea that, okay, this is something foreign. But each T cell is unique and can identify very different antigens. And this lock-in key mechanism is precisely what makes our immune system make targeted attacks only on your bacteria or viruses and not harm your own cells. This is for the enthusiastic immunologist who would like, this is how these are certain T cell receptors that are involved in detection of antigens. This is just a lot of jargon and we'll avoid that for the moment. So the T cells don't act alone. Once they detect this threat, they will multiply and create an army of identical T cells, and then they go and fight off this infection. Okay? So I'm going to show you a small video, and it's not just like fighting virus or bacteria, but also fighting cancer in our body. And I'll be just I don't know if I can show you this video, but maybe we'll come back to it. This video basically showed small gray T cells targeting your cancer cells and clearing them off. We'll come back to that later. So, are all the T cell receptors same? That's not the case. our body can produce at least approximately 3 million different T cell receptors, and each of them is very unique, correct? So it can detect a range of bacteria, viruses, and pathogens, and even abnormal molecules on our cells. But because there are so many different receptors, inevitably, some of these receptors can identify your own cells as foreign, correct? And this problem, these receptors identify something called as self-antigens. And these self-antigens, and this is called the self-reactivity problem. Now, what to do about it? Obviously, as nothing in nature is perfect, and nature has a solution for everything, our body has a very nice mechanism. Now go back and remember your thymus, right? I said it's like a training school for your T cells. So when your T cells move from the bone marrow to the thymus, okay, when they come out of the bone marrow, they are immature. They don't know what to react to and what not to. But in the thymus, they are presented with self antigens, the antigens that are present in your body. And when that happens, and if a T cell recognizes this antigen with a very strong affinity, or it's like a very strong response, then your body will kill this T cell and remove it from circulation. So such T cells never enter the circulation of your body. So this mechanism is called central immune tolerance, or central T cell tolerance. And the Nobel Prize for this was given way back in 1960s to Burnett and Medaver. And for 20 years, the whole field thought that this was the only mechanism to eliminate these self-reactive T cells from the body. But is everything perfect? No. Some of these T cells still escape this detection in the thymus and they move out in the circulation. So the question is, how does the immune system control these self-reactive T cells that have escaped the test in the thymus? So here comes our first Nobel laureate, Shimon Sakaguchi. He precisely asked this question, and he did a very elegant experiment. He took baby mouse newborn pups and he removed the thymus. Now, knowing that thymus is the training center, these pups would have a very overactive immune system. So they develop some symptoms of autoimmune diseases. So with this model, what he did is, he did something very elegant. He said, what if I give mature T cells from a healthy mouse to these sick animals? What will happen then? And surprise, surprise, yes. Newborn rat as small as millimeters. Yeah, 0.5 millimeter. It's that tiny. And for us, it is as big as your fist over here. But it keeps decreasing as you age. Okay. So, surprise, surprise, these mice became healthy. And that's when this was the turning point in history, which showed that there are certain T cells in your circulating immune cells that can prevent these autoimmune reactions. But this was not enough. Sakaguchi wanted to know which T cells. And at that time, scientists already knew that there are many different T cells, okay? And they used they had different function too. Like certain T cells would directly connect with the target and kill it, while others would activate, like signal other immune cells to mount an immune response. So scientists use something called as these markers, molecular markers again, which are on the cell surface of your cells, called CD markers, to identify different T cell subsets, correct? So think of them as like name tags, right? So each T cell will have a combination of different name tag that will help the immunologist to tell apart which T cell does what and what is their role. So precisely with the help of this knowledge, Sakaguchi went on the quest to identify which T cell subsets carry out this or prevent this autoimmune reactions. So again, he did a very powerful experiment. He took the same animals that became sick right after the thymus was removed, and he isolated this different T cells based on the cluster of differentiation proteins. So first he injected CD4 T cells in these animals, but these animals still remain sick. Then he added this another subset which also expressed CD25. And this, when both the combination of these cells was given, is when these mice became healthy again. This was the experiment that showed that CD4 and CD5 T cells have the ability to suppress your immune reaction, and then Sakaguchi went on to call them as regulatory T cells or Treg cells, right? But again, scientists are never satisfied. So at that time, although this CD4 CD T cell 25 subset was identified, what scientists found that there were other type of T cells that also express the same molecule. So the problem was we still didn't have a definite marker for your suppressive or regulatory T cells that will tell us that these are precisely the cells that stop the immune reaction. Here comes, sorry, this we have gone too far. Okay. Here comes the second breakthrough. Again, a lot of things in science happen as serendipity. And there were certain scientists in the Manhattan Project that were working on understanding how radiation affects mice. Okay? And while doing these experiments, they found a very novel mutation, which was called the scurfy mice. Okay? Now, the scurfy mice had a very scaly skin, and so the name scurfy, they nicknamed it scurfy. But there was something very interesting about this mice. Only the male mice developed this autoimmune disease, which gave scientists a clue that there is a mutation in your X chromosome that is causing this particular phenotype of autoimmune disease. So, this task of finding the gene that causes this autoimmune disease was taken up by Mary Brunkow and Frederick Ransdell at Celtech Cyroscience. They were working in this company in USA, and today we have very sophisticated instruments and we can do sequencing in just a week, whereas it took almost 10 years for the human genome to be mapped, right? And today we have the whole genome, but way back in 1960s when they started this work, it was a painstaking task. And yes.
Manhattan Project, probably there were no biologists.
Yeah.
So what made biologists look at the mice that way? Is there anything?
No, so they were looking at the effects of radiation, and they couldn't obviously put humans in those chambers and check. So they studies on this effects were done majorly on mice, although they were no biologists, but I think a lot of chemistry people also kind of look at this particular phenomenon, right? And so these people found that there were 20 genes on this particular section on the X chromosome, and one by one they started looking at each gene, whether you remove that gene, what happens, you put it back, what happens, and after doing all of that, they found that the FOXP3 gene is the one that was responsible for this scurfy phenotype in the animals. And that was not enough, right? It is in the mouse. It was just a novel mutation. What about humans? Do we have this gene in humans? And to kind of translate their findings to humans, they started working on a very rare autoimmune disease called IMPEX. And for the full form of which is immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. We'll not go into the details, but these children develop severe autoimmune diseases. Their immune system targets their pancreas. They have less number of blood cells. They have kidney damage. They have neurological problems. They have issues with their digestion and gut system. And obviously they have eczema and all. And interestingly, just like scurfy mouse, this autoimmune disease only happened in boys, which also gave them a clue that again the mutation will be only present on your X chromosome. And again, they took samples from these sick patients and they analyzed their genome, to which they understood that again the FOXP3 gene was responsible for causing autoimmune diseases like MPEX. This discovery definitively told the field that the FOXP3 gene was the master regulator of T regulatory cells, which then can act as or then can act to prevent your autoimmune reactions. So let's go back and see how much time this has taken. Right from the central tolerance being identified in the thymus to Sakaguchi's identification of CD4, CD5, CD25 positive cells, to identification of FOXP3 gene in the mouse, and then finding that gene in responsible causing and it causing autoimmune diseases in humans, it took almost half a century, 50 years worth of work. Now that we know what T regulatory cells are, and once this subset was discovered, other scientists came in and they started studying how they interact with other immune cells and what happens in different conditions. So now that we know how the discovery happened, let's understand a little bit more about Treg cells. So what do they do in the body and how do they protect, correct? So when any T cell detects your cell as foreign, you have these surveilling T cells in your immune system that will detect this connection as an error. And they will come and release certain molecules which are suppressive factors, a cytokine called IL-10. And this IL-10 causes your normal T cells or activated T cells to die or destroy themselves or not react to the self-antigen. Now, this is not just the role of T cells. The physiology of T cells goes beyond this. And suppose, say you have a viral infection, a flu. So what happens, right? The first 2 days you are kind of sick and ill, and after 3-4 days you become fine. So what is happening in the first 2 days? Your first 2 days, your immune system is now recognizing the virus, fighting it. So you have fever, you don't feel like going anywhere, or you have that mucus coming out, which is also an immune reaction to remove the virus from your body. But once the virus is cleared off, something has to tell the immune system to stop, right? Now enough, the virus is gone. You have to calm down and come back to the normal state. This is precisely what T regulatory cells do. If you look at your T cells when you're having such a small infection, in the first 2 days, you will see very high number of activated T cells and you will not see any T regulatory cells. But by the 3rd day or something, the levels of T regulatory cells start increasing in your body and they will then control the infection. If the threat is still there, they may not be be able to control the infection. So again, your immune system will keep fighting, but it will still be maintained in control. Now, this is again not the only role, and it plays a very important role in other physiological conditions like pregnancy. Now, the baby carries genes from both, right? A mother and the father. Now, father's genes are obviously foreign to the mother's immune system. So the mother's immune system is primed that anything foreign, please attack. So this is precisely where your T regulatory cells come. These T regulatory cells suppress the mother's immune system and allow the baby to grow safely in the womb. So they line the placental region and suppress the T cells that recognize these fetal cells as foreign. Okay? And similarly, how the way these Treg cells protect from the fetal antigens and provide tolerance to the fetal antigens. So one more thing I liked, and that's the precise reasons where mothers are asked not to fall ill. Because if you're falling ill, you already have a heightened immune response and that can harm the baby. So it's very important, although we have Treg systems in place, but a little bit of caution is always advised. Not only that, similarly, these Tregs play a very important role when someone is undergoing a transplant, whether it's a bone marrow transplant, liver transplant, or a kidney transplant. What happens? Although you do all the matching and everything, but each one of us is a unique individual. No two people can be exactly same unless, of course, they are twins from a same zygote. But other than that, most people are unique in the antigens they express. So when you have a transplant, there are some small antigens that may cause a mismatch, and due to which the patient may reject whatever is being transplanted, be it the bone marrow or the liver graft or the kidney graft. And exactly, it has been shown that if you have higher number of T regulatory cells in the system, you will have a better approval of the graft in the body. The body will be able to survive with the graft for a longer time. But is all good with T regulatory cells? Are they always the heroes? Not always. This is where a very interesting thing happens in cancer. Cancer hijacks this power of your Treg cells to suppress your immune system. Your immune system in generally can detect cancer and remove these abnormal cells from your body. But cancer does something very interesting in reality. What it's going to do is it's going to release some factors that is going to call all the Tregs to its microenvironment, right? And these T regulatory cells now, instead of protecting the body, start protecting the cancer, correct? And that is why today there is a lot of research where people are trying to develop therapies in which we can reduce T regulatory cells in the cancer microenvironment to target cancer better. So everything about Treg cells is not about having them higher or lower or just about providing protection. It's basically about the balance between your inflammatory cells and your T regulatory cells. If you tip the scale on either side, you're going to have a disease condition. So if you have to stay healthy, your body has to maintain this particular balance in your immune system. And this knowledge is what has led us to develop better therapies for diseases like autoimmune diseases, cancer, also to combat complications that are related to transplant. And that is why this Nobel Prize discovery has changed the way we see immunology. I will be now open to questions. Thank you.
That's still quite fast.
Quite fast.
Yes. So, all right. So what we'll do is
Yes, I can show that.
We'll open the things to questions, but before
Yeah.
that, we'll close the online part. And
Right.
so before we close, we wish to thank St. Xavier's College for accepting our request to host a Chai and Vai over here. And the next Chai and Vai will happen on the coming Sunday at Prithvi Theatre. It will be on Nobel Prize in Physics for the year. Okay. So with this, we'll stop online thing.
Yes.
And then the floor is open to the questions.
Right.
The video first, I think.
Yes, I will show all that. Just a minute. Because they won't be able to see the video online. So if you see this,